Hi everyone, my name is Gwen Joseph and I'm going to give you a really brief overview of my research project titled Hypoxia drives CD4 positive cytotoxic T lymphocyte differentiation. I first want to give a really brief overview of the characteristics of CD4 positive T helper cells. These cells play an important role in the adaptive immune system. More specifically, a CD4 positive T lymphocyte is a type of white blood cell that stimulates cytotoxic T lymphocytes, which in turn are cells that kill damaged or infected cells, typically cancer cells. Under normal body conditions, they release cytokines that help the activity of other immune cells. Under situations of chronic inflammation, however, they can produce cytolytic mediators such as perforin and granzymes, which induce target cell apoptosis in a cooperative fashion and can contribute to disease. Despite the importance of these cells, little is known about the factors that drive their differentiation. Now I want to briefly go over some experiments performed in Olson Lab that contributed to the formation of my hypothesis. Olson Lab primarily focuses on graft versus host disease, which is an immune condition that occurs after transplant procedures when immune cells from the donor attack the recipient tissues. Our first step was to determine what some key characteristics of graft versus host disease are. Through using lethal irradiation to knock out the immune systems of mice, and then implementing either a syngeneic or allogeneic transplant, we were able to determine that granzyme A producing CD4 positive T helper cells are found in graft versus host disease. Through further study, we found that the presence of granzyme A positive CD4 positive T helper cells is required for the development of lethal graft versus host disease. When these cells are not present, the mice had higher rates of survival. We cultured naive CD4 positive T cells under various cytokine conditions to determine which combination of cytokines led to the highest expression of granzyme A. Using these results, we then performed a genome analysis to identify genes highly expressed under these conditions, or these conditions when granzyme A is highly expressed and therefore genes that may have a relation to graft versus host disease. Hypoxia-inducible factor 1 alpha, or HIF1 alpha, was flagged, leading to our hypothesis that hypoxia-driven HIF1 alpha enhances granzyme A production by CD4 positive T cells. To test this hypothesis, we used an experimental design centered around harvesting spleens from wild-type mice and isolating naive CD4 positive T cells. These cells were then cultured under various cytokine combinations and then under either normoxic or hypoxic conditions for 5 days. On day 5, the cells are harvested and analyzed through intracellular staining and visualized using flow cytometry. Before I talk about the results, I want to first give you some information about what is typically seen in cytotoxic T lymphocytes. Granzyme A, granzyme B, and perforin are all upregulated and function in assisting cytotoxic T lymphocyte-mediated apoptosis of infected cells. Cytotoxic T lymphocytes also typically have low TH-POC expression, which is important in maintaining TH cell identity, and high CD8 alpha expression, which is important in intercellular interactions. Our results show that granzyme A and granzyme B expression are upregulated in hypoxic conditions, and more specifically in the TH granzyme A cytokine condition previously identified, as well as in the TH0 or nil condition. You can also see that we observe very little perforin variation and between normoxic and hypoxic conditions. Furthermore, you can see that the cells high in granzyme expression do not exhibit the classic cytotoxic T lymphocyte phenotype and instead have a very low CD8 alpha expression and a high TH-POC expression, suggesting that there is a possibility of a novel subset of cytotoxic T lymphocytes. I want to briefly summarize the conclusions that can be taken from this data. We first determined that hypoxia drives granzyme A and granzyme B production in TH0 and TH granzyme A conditions while not driving the production of perforin. We also see evidence that hypoxic conditions do not induce a classic cytotoxic T lymphocyte phenotype as seen through the CD8 alpha and TH-POC expression levels. Work that I am currently performing to further this project is centered around modulating and quantifying HIF1 alpha expression through real-time PCR to determine if it has more of a sufficiency or a necessity role in graft versus host disease. Lastly, I would like to thank and acknowledge members of Olson Lab who have helped me throughout my undergraduate research journey.